This is the Shahid Shah Show. Straight talk on building, buying, and investing in digital health. No hype, no buzzwords, no speculation. Just what actually works, maybe what doesn't, and most importantly, why. If you're building healthcare technology or buying it, this is where reality shows up. I want to tell you about a biopsy that never happened. A patient has a tumor sitting close enough to a major vessel that interventional radiology looks at the scan, looks at the referral, and declines. Not because anybody's being careless, because sticking a needle in that spot is genuinely a bad idea. So the tumor board waits. The oncologist waits. The patient, who does not have unlimited time to wait, waits the longest of all. Now, we've built a system where a tumor is often more knowable than it is reachable. We can basically characterize a piece of tissue down to the transcript level, but if nobody can safely get the tissue out of the body in the first place, all of that capability sits on a shelf unused next to a very, very expensive sequencer. So this is part two of our three-part series with Caris Life Sciences. If you're here for the first episode, you'll remember James Hamrick and I spent about half an hour on whether reading a tumor's entire exome and transcriptome instead of a curated panel actually gets the right treatment decision made faster, or does it just hand the oncologist yet another long report that they may or may not be able to make a decision from? That was our in-depth question. Today, we're going to reach another question, and the question is, it doesn't matter how deep your test is if nobody can get to the sample onto the bench in the first place. So what do we do in those cases? So for about a decade, the industry has promised a fix for this. It's called liquid biopsy and a blood draw instead of a needle. Every conference has a slide on it. Every slide said it's about to close the access gap. I've been hearing about this for a while, and certainly my guests have as well. And so today I wanted to really pressure test that promise and not just talk about the future. Caris Assure is a pan-cancer blood-based test built on a whole exome and whole transcriptome sequencing of circulating nucleic acid. And it's one of the few in this category that really looks at both plasma and the Buffy code, as well as a bunch of other technical stuff that we're going to talk about here. And that's a mechanically different claim than we found DNA in the blood, and we want to know whether this holds up. So the question for this episode, and we wanted, just like every other episode on the Shahid Shah Show, you want to penetrate deeply, ask partners and vendors deep questions. And today, that question is about liquid biopsy. How good is it? Of course, it exists, but the question is whether it's actually closing the access gap. So joining me today is Dr. Joseph Murray, Senior Medical Director at Caris Life Sciences. He's a physician scientist by training with a commensurate MD and PhD in tumor biology from Georgetown, residency and fellowship at Johns Hopkins. And before Caris, he co-directed the Lung Cancer Precision Medicine Center at Johns Hopkins, where he spent years integrating this large-scale clinical and genomic data. And this is going to be a very, very fun conversation because he's built an academic program from the inside and now sits in a commercial one where not all of the stuff that he's worked on in the past, which just had to go into papers, can stay in papers. They have to go into patients, and that's the exact vantage point that we want to show. So, Joseph, with all of that intro, welcome to the show.
Jay, thank you for the kind introduction, and I'm happy to join you today on a topic I'm truly passionate about, which is liquid biopsy.
Yeah, and the work that you've done in the past, has a lot of value today. But I think the fear is that a lot of people don't really understand that. So let's start with the mechanical side. Just tell us, what is circulating nucleic acid sequencing, and how is it actually different from the ctDNA only liquid biopsy test that a lot of our audience might already be familiar with?
Certainly, circulating nucleic acid sequencing involves something unique to Caris, which is the simultaneous analysis of cell-free nucleic acids. This would be both DNA and RNA from blood. And so this aligns with a similar approach we've taken with our tumor tissue testing, where we conduct simultaneous sequencing of DNA and RNA from tissue as well. And this is our aim and goal is to gather as much information as possible from every sample. Patients, as you mentioned, go under various biopsies at times, and we want to take as much value and information comprehensively from those specimens collected. We focus on capturing both cell-free DNA and RNA from the blood to complete whole exome and whole transcriptome sequencing. These currently are validated for circulating nucleic acid sequencing for our Caris Assure product, but I will comment that the reports are really therapy associations primarily identified from the whole exome sequencing data. However, collecting this comprehensive whole exome and whole transcriptome sequencing from circulating nucleic acids is a cornerstone, and as a whole for every specimen and each patient enables the design and evolution of future versions of our assay, and really speaks to that completeness that we aim to get with every specimen collected.
Nice. And so why look at both plasma and the Buffy code? What specific problem is that solving that plasma alone doesn't? Because we've had a plasma alone for a while now.
Yeah, and in differentiating between circulating nucleic acids, now getting into the space of circulating tumor DNA, I'll start by saying that plasma-only liquid biopsy sequences all the cell-free DNA with the goal of identifying exactly that, variants in circulating tumor DNA. However, the plasma is flooded with cell-free DNA from various sources in the body, and this includes normal organs and tissues. Variants can occur in any one of these organs and tissues over time, either those that we're born with, which we call generally germline variants, or those that occur over time naturally in organs. For example, a process called clonal hematopoiesis that occurs in the bone marrow of some patients, and the bone marrow that produces white blood cells. So those can spill over into the bloodstream as well. So plasma-only liquid biopsy can't really differentiate the origin of a variant in this way just by focusing on plasma-only sequencing, which is where paired plasma and Buffy coat sequencing truly shines. It's the gold standard for identifying the origin of a variant found in plasma. And this has real and practical clinical implications in my eye. Pathogenic variants in specific genes, say in prostate cancer associated with therapies, are indicated if a patient has a germline or a tumor mutation, but not one from clonal hematopoiesis. And so this is an actionable place where the data from our assay can drive a clinical decision and hone therapy to a patient more specifically. So, in my eyes, getting that origin of a variant is an absolute, and it truly can benefit a patient.
Yeah, let's double click on the origin part again because in our last conversation in episode one, that did come up again. And especially today, when there are hard to reach tumors, what does this process give us about the origins that we couldn't do with just a regular biopsy?
Yeah, this is where, when we're sampling such a diverse space in the blood as well, we can pick up on something I think you're alluding to, which is tumor heterogeneity as well. Sometimes we have, what looks like an easily accessible biopsyable spot, but in a patient with metastatic disease, they may have multiple sites that are less accessible to sequencing as well. And so, by doing a plasma and Buffy coat comparison on a liquid biopsy, we can really try and identify where we might find, yes, tissue specific variants we've already found, but also new insights into other mechanisms of potential resistance that are tumor specific by excluding things that are germline or clonal hematopoietic events. Really reaching and speaking broadly about heterogeneity that actually happens in patients.
Yeah, there's really a whole bunch of cool things that can be done now. So if you want to give maybe a few practical patient examples, and then talk about where this works best, but where it's got limitations that physicians and prescribers should know about.
Yeah, I'm keen on liquid biopsy as you know, but I do want to comment on several limitations there too, even with the most sensitive assays we can develop, right? Tumors are inherently different in their biology, in their localization, and just their characteristics. And we focus a lot in speaking about liquid biopsy on the ability to detect nucleic acids from a tumor specifically. And this relates to a process we call tumor shedding in general. Um, some of the you and some of the folks on this call may be aware that there are certain types of cancers like colorectal cancers that are higher shedders on average than other cancers like brain tumors, which have a lot of other barriers where circulating nucleic acids could get to the bloodstream. But even within the same organ, let's use an example near and dear to my heart, which is non-small cell lung cancer. Squamous non-small cell lung cancers tend to shed more than adenocarcinomas, where we actually really do want to identify actionable alterations in that latter group. Now, tumor shedding is also affected by volume and kinetics of cancer, and these are things a patient may be feeling and experiencing, right? Large tumor volumes causing symptoms, rapidly growing cancers that are increasing symptomatology over time. And this is where cancers will shed more versus stage one cancers that are small, localized, and often asymptomatic, right? Um, and there's no liquid biopsy, I think, that's perfect to address this. We can try and enhance tumor shedding through novel ways, but really the goal is to get as much diagnostic information from the tests we have on hand. And so, to give an example, like you're saying, I think about resectable lung cancers. This is a space where actionable alterations are becoming more and more important in patients' care journey. And tissue-based testing, is the standard. Using a liquid assay to rule out an actionable therapeutic target in an early stage lung cancer is incomplete. A negative test does not mean there's nothing to target. Rather, we're up against the limits of things like the biology of the cancer, the stage of the cancer, and this tumor shedding concept, and the assay as well, just to be clear. And this is where tissue testing is still mandatory.
Got it. If you're just tuning in, this is the Shahid Shah Show, where we cut through the noise in digital health and talk honestly about how healthcare technology actually gets built, bought, deployed, and paid for. Today, in part two of our three-part series with Caris Life Sciences, we're asking whether liquid biopsy has actually closed the access gap in precision oncology or just added another test to the order set that people have to go through. Joe, you've taught us a lot about the liquid biopsy part here. So let's walk through how blood-based profiling actually changes a decision for a patient who really genuinely cannot undergo a tissue biopsy because of location of tissue, et cetera. And maybe not the hypothetical patient, but do you have one that maybe, obviously you can't identify one, but a patient that you've worked with or have seen in real life benefit from this procedure?
Absolutely. I've certainly used blood-based profiling in the diagnostic journey of many of my patients, particularly in cases like you mentioned, where provisional tissue biopsies give us a diagnosis. Maybe they tell us it's lung cancer, but they're insufficient for molecular testing. This is unfortunately a common scenario, and we have this term quantity not sufficient or QNS as the tissue challenge here, where liquid biopsy can augment. And so let's consider one of those scenarios. I had a patient generally that I took care of, never smoker, lifelong quite healthy, but suffering from dementia and who presented with a cough, shortness of breath, and findings of a lung mass. They were hospitalized at the time and they really struggled with both dementia in the hospital and getting through all of the therapeutic or diagnostic journey that was needed. Even with the best intents and the best biopsies and all the tools in the hospital, we had a QNS, you know. And in shared decision making in the outpatient setting, the patients, back in their home environment with their caregivers, proceeding with a repeat tissue biopsy is not a realistic option in the scope of care. And so a liquid biopsy in that setting, in this setting yielded an actionable EGF alteration and starting an oral therapy quickly based on that biopsy result, be it liquid or tissue, really led to their improvement. They had rapid improvement in clinical symptoms, their overall well-being, and it just matched meeting the patient where they were. So it was the right test at the right time.
Yeah, that's almost a perfect example. So let's maybe dive a little bit deeper into the case for running this tissue and liquid concurrently instead of just picking one. And you mentioned time as one of the important factors. So is it actually faster to get to the treatment decision or, a fuller molecular picture or something else? If you can walk us through do when to do both, when to do one, and how does it actually change the pace or the speed of the decision making, which of course is the most important thing that these things are supposed to let us do.
Yeah, absolutely. I will start off by saying I'm biased. I'm a strong advocate for comprehensive molecular testing for every patient, and I'll use metastatic lung cancer as an example here. As we discussed, a tissue diagnosis is required for a cancer diagnosis. So we often have a saying when tissue is the issue, when we're searching for cancer is the answer, right? And so this is founded on the fact that independent of our most comprehensive molecular testing, something I'm an advocate for, tumor agnostic approvals aside as well, histology and cancer type still primarily differentiate clinical care, right? So getting that tissue biopsy is absolutely critical. Whether you can get molecular testing is the question here, and this is where concurrency matters. We don't know the end results of these pathways of getting liquid and tissue biopsy up front. We only know that we can implement both, and I'm a strong advocate for doing concurrent testing to get the answer as soon as possible clinically for care. There is published work out there that suggests this is the case, and we're doing our own work to validate and verify this information. So molecular testing from tissue is a natural next step because it's there and available, but it may not be ready for all the analysis we need. I'll say in lung cancer, though, there's still IHC-based testing for biomarkers that are highly relevant in first-line care of lung cancer patients. So having that tissue is requisite and required. It's a necessary part of the journey as we get all the additional comprehensive molecular information. And I discussed in that prior clinical scenario, tissue may be limited or unavailable, or it may be pending at the time of a visit I have with a patient. And that's where I still use liquid biopsy. If it's going to get me an answer faster, this will reassure the patient and get them on a treatment plan earlier. There's another part about this comprehensive question that I want to clarify. And you kind of commented on like the full molecular picture of what's going on. Beyond timeliness at the time of diagnosis, concurrent testing up front gives us at that time everything we can know if we do it comprehensively. That cannot be repeated at every time a patient may progress on a treatment, right? And so having that information locked in time as comprehensively at the time of diagnosis is needed. It may inform future directions of care when we can't get biopsies or we're limited in tissue testing for IHC-based biomarkers beyond the things I want to find in liquid biopsy, like variants that identify resistance. And this is baked into how we develop our products too, in thinking how we track things over time in tissue and liquid testing, be it concurrent or sequential over time when we're monitoring, or how we use things like look back programs to identify patients who have prior molecular testing that is revealing for new FDA approved indications as well. And so I think we integrate this well. I think about every patient benefiting maximally in their journey from the most information. And that is a bias I'll have as a physician scientist. I'll call that out. But in thinking about how we can use tracking over time and look back programs to improve care, this is why. This is the reason. There is a clinical reason we include these things in our reports and in how we engage with oncologists and their patients.
Yeah, in our last episode, we talked about this problem of what I've talked to my audience about called decision debt, that sometimes you get so much information that you're sitting on it and then you don't make a decision. And now this debt keeps piling up and interest is being paid on that debt because you haven't made the decisions to go execute on something. You're just looking at more information. So what I really found cool about that, I think you guys call it the track table and the look back program. So could you talk a little bit about maybe from your own research or clinical leadership, if in a real world case, did something like with the track table or the look back program change, after you made a decision, then you got some more information and then you changed the decision. That might be an interesting anecdote to hear about.
Absolutely. I think starting like steadfastly biased that comprehensive testing is needed. Let me characterize this with a story from a patient with a very aggressive non-small cell lung cancer. And this is a patient who underwent testing when I was still in clinical practice and had not joined the company yet through Caris. My patient was very symptomatic from the start, lots of issues with breathing and pain and other symptoms. And I did, as I normally do, I ordered concurrent liquid and tissue testing from the start. And the former identified actionable EGFR and P53 mutations up front, and that meant I could start a therapy, even while the tissue testing was still pending. And that tissue testing, though, gave me more information. It revealed the same two alterations, but also a pathogenic alteration in a gene called RB1. And seeing this clinical scenario unfold, in all the genomic data that I got alongside, it really gave me pause in how I should think about this clinical scenario, what this meant for the patient with all this information, even though I knew I could start a therapy and get things going. And the reason is these alterations, beyond those that have therapy associations, do affect patient outcomes. And we know this historically based on retrospective studies and case series and whatnot. And that's where I understand like the decision debt of what to do with all this information. It's highly variable based on the hands of who has that information. And this is something that we find really important in reporting, hence things like the track table. And going back to my patient, she had a symptomatic benefit to targeted therapy up front, but, pretty quickly developed symptomatic progression again. She had fluid around her lungs and new lesions in the liver. and quickly dealing with symptoms first, pulled fluid off the lungs, and it said, "Yep, this is still non-small cell lung cancer," which I had figured, but it was insufficient again for testing. So I got that comprehensive concurrent testing up front, but now I was in one of these scenarios where things are happening rapidly and I need more information. So I had also sent liquid biopsy at the time, and it revealed a new fusion, an FGFR2 fusion, which is an emerging biomarker in lung cancer. And I was excited about this, honestly. But it also noted that not only like before the EGFR and P53 mutations were, but the RB1 was present in the blood now. It hadn't been before, but now it was. And this speaks to the idea of what is really driving the progression here, what biology is happening underneath. This made me think more about small cell transformation in this patient. And so I know this is getting in the weeds, but this is the practical nature of how we think about metastatic lung cancer care, even with these, fancy therapies for driver positive disease. This patient, under liver biopsy had small cell transformation. Disease was acting differently, heterogeneously at different sites. And so if I had just started that FGFR2 targeted therapy and said, we're great, we're good, everything's going to respond, I would have been missing the mark because small cell transformation needs to be treated differently. So I recognize this may be a maybe a molecular case that's very specific and detailed and involved. But if we're not thinking clinically, very like honing in on what's happening to our patient and recognizing the limitations of our assays, we can't also take advantage of their comprehensiveness. And so I think this one truly exemplifies the need for that comprehensive care backed by comprehensive testing. I think those go hand in hand.
No, I think that was a perfect example of the complexity. I mean, the weeds, you have to be in the weeds in these cases. So my mom actually has gone through almost, she could actually be your patient that you're mentioning. So in these same scenarios, minus the dementia, she's in good shape mentally. But this same exact issue we've talked to multiple oncologists, they tell us things, but then they say, based on what I know from a month ago, the imaging or the tissue, and that's scary, right? A month ago, I mean, you've got her on Tagrisso for 4 weeks now. What can we know from a month ago versus what can we know from today? And so that timing was very, very important. Now, what we've seen though is that as we talk to oncologists in different places, there's a real barrier to the adoption amongst clinicians, of course. Some of them are skeptical because, they just don't understand the technology. Some of them are skeptical and like, look, I understand the tech and I get the report. I haven't been through any of this training that you guys have. What do you want me to do with it? So, how do you talk to physicians who are legitimately skeptical about this stuff on what they should do to get maybe, is it called upskilling, side skilling, whatever you want to call it?
Yeah, and oncologists are inherently skeptical because I think physicians are in general. And I do believe, though, they're becoming less skeptical around blood-based testing in general because patients respond well to it. It's less invasive. It can be done in a clinical setting that's easy, quick, just like a lab draw. And so there's this balance of skepticism, but also adoption. And in some ways, I worry about more oncologists trusting blood-based testing alone, as I don't think it reflects comprehensive molecular testing for all patients, as we discussed in a couple cases. And so I just want to talk about where maybe that skepticism should remain. I think there should be a healthy skepticism of a negative liquid biopsy result. That's absolutely needed. Liquid biopsy assays remain challenging with sensitivity and the tumor biology. So, although we've enhanced our assays over time to deal with sensitivity, the challenge is as cancers evolve and respond to therapies, but then progress, we may still be at those limits of detection. And so I hear the skeptical and practical oncologists say, let me start with just the tissue then, and if that's the most sensitive. And in a way, it's harkening back to the adage I mentioned: if cancer is the answer, tissue is the issue. And so this is still a comprehensive approach to testing. You can still pair the two, but please be skeptical of a negative liquid biopsy. And I think there should be also a healthy skepticism of a positive liquid biopsy that just has an overwhelming amount of information. Liquid biopsies that report really sensitively at the extremes of assay sensitivity are also prone to false positives. And those can drive, not to be too pedantic about it, but they can drive false hopes, right? Ideas that there is a therapy that matches. And so beyond these false positives, knowing where a variant comes from, we spoke about that a bit, and putting it in the right, what I call diagnostic bucket, really speak to identifying, where liquid biopsy can augment and improve at the levels of the most sensitive assays. Even when things are positive, we need to find where positivity lies. Is it in clonal hematopoiesis? Is it really from the tumor, or is it something the patient was born with? And so this is being comprehensive too, putting things in the right label and category, and really identifying where signal is greater than noise there, and identifying lastly how we can link all that to the therapy. That's what matters for the patient. So in the end, our goal is to empower physicians with the right set of tools that balance skepticism with practical evidence, right? We really want to be practical about this. And that's what's going to enable the best care of patients. Just like you said, we don't want decision debt. We want to be able to enable, physicians, not just physician scientists, to really push the boundary of taking test results and aligning with therapies that benefit patients. And so that'll
Perfect.
forever be my passion in liquid biopsy and beyond.
No, no, this is exciting stuff, and just as a caregiver to a patient that has gone through this, these kind of things that you guys are working on are literally life-changing. So awesome there. Just one side note before we conclude is that on the business model side, a lot of our audience members would like to know like how much does this cost, how does it get reimbursed, etc. And I'm not going to put you on the spot for that because reimbursement policy is actually its own specialty and it changes a lot. But just for our audience to know, liquid biopsy today mostly gets billed as an add-on alongside the tissue testing, not a replacement for it. And so untangling that through the payers and how you handle it, it can be handled through your normal workflows and your normal EHRs, et cetera. So you shouldn't have to worry about saying, oh, do I have to know something different? So I wouldn't worry about that. But the business model is important to know because obviously these things have to get paid for. And with that, thank you so much, Joseph. That's a very, very honest picture of liquid biopsy than most of the things that we've seen out there. And for the audience, next time we're gonna do a final part to this series, where we're putting a million molecular profiles on the table and asking what a data set that size is actually good for once we set the word AI aside. And as always, this is the Shahid Shah Show. And until next time, keep innovating, but stay real and practical the way Joe showed us today.